Hello, everybody. I'm Alexander Maxon, research lead at HSC, and today I'm introducing HSC's Research Council and their interactive session, Ask the Scientists Anything. The Research Council of the Huntington Society of Canada is composed of the following leading HD research scientists: Dr. Rachel Harding, Dr. Tamara Maiori, Dr. Tiago Mestre, and Dr. Mahmoud Pouladi. Council members are volunteers that make recommendations about applications received for HSC's research funding programs, such as our Navigator Award. The Research Council also provides guidance regarding the Society's role in funding HD research, which includes scientific advice about specific research projects and proposals that may be funded in combination with other national and international funding bodies. The team will be answering all of your questions about HD research, including clinical and observational research and studies. No question is too simple or too complicated, so feel free to ask whatever you've been waiting to understand. For a couple of housekeeping items, you can ask your questions verbally to the panel by raising your hand here in Zoom. When called upon, you will be unmuted and then you can ask your question. We will also be taking questions from the Q&A tab on FeedLoop if you prefer not to ask verbally. We will get to as many questions as possible. Unfortunately, due to the volume of questions, we may not be able to get to everyone. So please try to make sure your questions are not too specific to your situation and are instead general so that they are relevant to the broader group tuning in. So let's get started. So if anyone would like to get started, the first question I know somebody has asked was they'd like to know more about stem cell therapies and if they're an option for replacing dying cells in the brain. If anybody would like to take that.
Mahmoud, do you want to field that one?
So that's a great question, and I think there's been a lot of interest in developing cell replacement therapy, essentially ways to grow cells or to implant cells to make up for the ones that degenerate in disease. So it is something that is a possibility. It's not really, I would say, close to being deployed in the clinic. It's something that's still under active research and development. There is a global effort, not just for Huntington's, but also for other neurodegenerative diseases. And there are many efforts around the world that are focused on doing this specifically, essentially either using neural stem cells or using neurons, essentially the more mature forms, and implanting them into patient brains to see whether or not they could help. But we're not really there yet, and so it's not something that that's likely to happen in the next 2 to 5 years, but likely sort of to be in a longer time. So I would say essentially something to monitor. It is a possibility, and we'll be monitoring essentially the research to see how far and how quickly it develops.
Mm-hmm. So if that's stem cells, another technology that was often discussed today and people were asking questions about was CRISPR. So is there anyone who could go a little more into detail about specifically what CRISPR is, how we use it to edit genes, and what we can use as a potential treatment option in the future?
Okay, Rachel, anybody? I'm happy to take this, but unless somebody else. Oh, Rachel is muted. Okay, so maybe I'll take this this question. So CRISPR is basically a defense mechanism that was discovered in bacteria. It's a way for bacteria actually to fend off against other pathogens that attack them. And scientists essentially were able to study the system and learn how to repurpose it for genome editing and genome engineering. And in a nutshell, we have DNA damage that our body has to deal with on a constant basis, and generally this DNA damage is repaired using a template within our genome. So we have 2 copies of every gene, and so if one gene is damaged, we use the good copy to fix the mistake or the damage in the other copy. And so essentially DNA damage can stimulate repair. And so the way CRISPR is used in genome engineering is that we can decide, or we can design CRISPR to go and target a specific gene in our genome to stimulate repair at that locus, at that region, at that place in our genome. And so let's say in the context of Huntington disease, we can use CRISPR to direct we can essentially use CRISPR to induce DNA damage at a very specific locus as a way of actually stimulating our bodies to repair that locus.
Mhm.
And this is how it's generally used, both in the lab as a tool to study what different DNA, what different genes and the DNA variation does, but also it's now actively being used as a way to treat cells. And this is, again, by either repairing mutations that we inherit or by modifying cells, again, that are isolated from patients in the lab or in the clinic, and then reimplanting them or replacing them. And I think Dr. Levitt this morning mentioned thalassemia as an example where essentially something like this is being done. The cells are taken from the patient, they're edited, and then put back in the patient. And it's under clinical development. It's not something that's approved, but there is definitely a lot of exciting progress happening in this area. And so it is something that's also being considered for Huntington disease.
I would just add, that I think the point you made, Mahmoud, is really important, is that right now, most CRISPR treatments that are kind of ahead in the kind of trials and everything else are all on mainly blood disorders, right? Where you can take the blood or other cells out of the patient, edit them, and then put them back. And so what remains to be done for neurodegenerative diseases or something like Huntington's is they've got to figure out, well, how are we going to do this throughout the body? Because we don't want to be taking people's brains out and putting them back in again. We need to come up with some better technology for how we might do that and do it in a way that's reliable. So something else that you might have read about CRISPR is that it is meant to be very, very precise, but there's still some issues with off-target effects sometimes. And the changes that are being made are irreversible, right? So we don't want to be using a technology that's still way too early in its development to edit someone's genome forever if we're not sure what kind of other effects we might be having with that. But these are all problems that there are so many labs working on this around the world, and they're all trying to make a better and better, more refined CRISPR technology system that can be implemented in that way. So as Mahmoud was saying, it's like important. We'll keep an eye on that, and hopefully we'll have some more exciting news soon.
Okay, so we've got some questions that are starting to pour into the chat here. So I guess I'll just go from the top down to the bottom. The first question is, has there been any studies done on cancer in Huntington regarding any effects chemo drugs have on those that have Huntington's disease at all?
I can touch on that, and then maybe somebody else will have more to add, but there's actually a reduced rate of cancer in the HD population. So that's something that's very interesting, and we don't know why exactly that is, but we don't have the information just yet about whether chemo drugs would be helpful for HD. But what we do have is a huge repository of information through Enroll HD. So a researcher could ask the question, out of the tens of thousands of people that are enrolled in Enroll HD, what drugs are they on? And there's going to be a certain proportion of people that have been treated for cancer, and what effect that might have had on their symptoms. So that's one way of getting at it.
Mhm. Perhaps I would maybe just throw two ideas. So I think one, when we look at epidemiology of cancer in Huntington's, you have to remember the age groups, and so if those studies were done matching the age of the participants, then it's interesting finding, right? As we get older, the likelihood of having cancer is greater in general. But I think also it links to more recent research in Huntington's, about DNA repair genes, right? And those are related potentially to cancer. And so as we look at those, and I will not extend much in that conversation, but basically they've been looking at how these DNA repair genes can be associated, for example, with the progression of disease or the time to the emergence of clinical symptoms, for example. And so potentially that could be a biological link of a lesser or greater risk of cancer for the individual patients with Huntington's. So that's an interesting thing.
So there's been lots of talk about inflammation this weekend. Is there anything the community can do to combat inflammation? How does it relate exactly to HD?
So
Anybody?
Diego, feel like this might
I can maybe comment with two aspects. So first, actually, there's clinical trials looking at. There's been and there's more recently a couple of clinical trials looking at modulation of inflammatory molecules. So molecules that we think are related with the worsening of inflammation are in the body or in the brain. And so one looking at this molecule called semaphorin 4, another one an inhibitor of a complement, which is just a inflammatory cascade of molecules that is related with any type of inflammation. And the results were mixed. So one didn't show any positive results, at least in motor outcome, but the one with the inhibitor of one of the molecules for the complement did show interesting positive results that potentially will lead to more confirmation of can we, by modulating inflammation in Huntington's, change how the disease progresses. But this question of inflammation is something that it's asked in other neurodegenerative disorders that at the first glance they don't seem to be related with inflammation, such as like diseases we call that are really with inflammation, for example, such as multiple sclerosis, which seems to be beginning. We know that's related with abnormal inflammation in the brain, but in other neurodegenerative disorders, there's always a question of is there primary mechanisms or it's just a reaction of the brain to ongoing neurodegeneration. But still, even if it's the second situation, it can open itself to a potential way of acting on the disease, because we know that excessive inflammation can potentially lead to more neurodegeneration as a self-feeding process. So I think that's one of the rationale why these different companies are exploring modulation of inflammation as a treatment for Huntington's.
I would just add that something that's interesting about the inflammation that we see in HD patients, it shows that Huntington's is a systemic disease, and we often think of it as being just in the brain, but it really does affect so many different organs in the body. And that's why there's so much interest in these drugs that might target beyond the brain, right? Because we don't want to be treating just the brain. That might be enough to start, but we need to develop drugs that are going to treat the whole body as well. And that's why there's more and more work being done in that area to understand Huntington's effects on the, things that happen to the heart or the liver, the pancreas, all these other organs that are really important.
Yeah, and maybe just one final comment on neuroinflammation in HD. There was a trial using a compound that, at least at the time, the understanding was that compound worked by modulating inflammation. And this was a trial by Teva Pharmaceuticals. The compound was laquinimod, and this was in the context of the Legato HD. trial, and the trial did not meet its primary endpoint, but it did. I think there were some reported improvements in brain volume loss and some other secondary measures. And so I think there is some clinical evidence that moderating or modulating the inflammatory response in Huntington disease can be helpful, but in terms of which aspects of HD and when to treat, and also whether there are maybe more effective ways of moderating this increased inflammation in HD, these are really all areas that would need to be followed up on. But it's clear that there is an inflammatory component in HD, and that potentially it might be driving some of the clinical features in the disease.
Great, thank you. I see a question here from Crispin. Hi, Crispin. Crispin wanted to know, how much have observational studies expanded, such as enroll HD, since first starting? And are these data that are gathered from these used for clinical trials, but are they the basis for the ones that are currently ongoing?
I just heard the last part of the question, might be just my connect, but I understand is observational studies use the data for clinical trials? Yeah.
Yes.
I maybe being more clinical, I definitely can say that yes, in different ways. So knowing more about the natural history of Huntington's allow us to, for example, address questions of who might be the best candidates for a specific drug. And of course, know more potentially, especially if we use imaging and wet biomarkers, try to understand more what's normally called like the endophenotype of a disease. And so there's that element, and there's still a lot of things to discover regarding that in Huntington's. But if you look at a study such as enroll HD, actually, if there's any of the participants are here today are part of the enroll HD, you'll know that the study itself, it takes the format of a clinical research platform. So not only participants are providing data that altogether will inform that better the natural history of Huntington's, but actually they can be candidates for other studies, namely clinical trials, and some of clinical trials that have more recently been ongoing. Actually, it allows not only for the pharmaceutical company that wants to know what are the most promising sites, but for example, myself being a site PI of enroll HD in clinical trials, even for me was very useful, because very quickly you have access to the participants at your site. You have access to data that again allows to link to a specific study. So allows for a more expedited recruitment in clinical trials.
And just to touch on the part about how much they've expanded, I think it's safe to say that it pretty much exploded. There are so many participants, which I always marvel at. It's just amazing how many people are willing to, make the drive to the clinic and take the time to do it. And it's hugely valuable. So to all those that are participating, thank you.
While we're talking a little bit about enroll HD, I see that Corey asked, is enroll HD still accepting new participants for all the categories?
Do you want me to address that question?
Sure.
Yes. At least on currently activated sites, it's ongoing recruitment, and so in principle, all usually I say it's a study for everyone in the HD family. So it's quite open to participation. I think definitely, and I think we need to be transparent about it. There's perhaps And I think it's, in a way, how the disease progress is that more likely people that are early in the disease will have the ability to take part in the study. It's still, it's still an extensive assessment that are done, although just annually. But in principle, it's open to everyone. So as I say, people that are gene carriers, people that from the same family that are gene negative, even spouses of individual with HD can be part of the study.
Thank you. I see a few questions here that are actually, let's start with this general one. So Deborah wanted to know, do we know when and where Huntington disease originated from?
Oh.
Yeah. Is there anybody who'd like to take that?
I can start it, and then you guys might know more, have more to add. But it's my understanding that the gene can actually kind of crop up. So basically, we have this CAG expansion, everybody has it. Like most people have maybe 18 or 20 CAG repeats. Some people have more, but they still don't get HD. They have closer to 30 or 35. Then they might get HD before, the end of their lives, or maybe not. But when you start to get up to that, like 37 to 40, I don't know the exact numbers, let's say 35 to 37, that's where things get a little bit variable. So it could happen that someone's 35 CAG repeat expands in their egg or their sperm, and then their child has an expanded CAG repeat, and that would be an example of HD cropping up kind of spontaneously. So does anybody want to correct that or further inform it?
I think you covered everything there, Tam.
So there's a couple questions here in the chat that people were wondering about other diseases in HD. Like, is there research being done? We've already talked a little bit about cancer, but some people are here wondering about ADHD, and they're wondering if maybe the medication they take for ADHD could possibly have an effect on their HD symptoms.
Mm-hmm. Should I have a go?
I think we need
Please.
the clinician's expertise on this one.
Yeah, I mean, I think that kind of leads to that still not fully uncharted area of treating symptoms in Huntington's, right? And likely you've heard during the conference about novel treatments, and even myself, when we look at treatment development for symptoms in Huntington's, I get always a sensation of frustration that really we've been able to find good drugs for chorea, but I think many of us know, and especially in families with Huntington's, that perhaps that's not the biggest problem in people with HD. And so currently we have at least 2 drugs approved in, I wouldn't say in Canada, but for example, in US, there's a 3rd one that will be, I think, soon available. And all of them do kind of the same thing. So they deplete dopamine in the brain. But then if you look at other areas that perhaps are more important in terms of how they impact quality of life, and so you mentioned ADHD, but I would put it that in behavioral domain, irritability, apathy, and then issues with memory. These are still things that we have not found good drugs that specifically show the benefit in people with Huntington's. And so what we try to do as clinicians is use the best evidence in other conditions, or for example, depression. We will use drugs that either myself being a neurologist or psychiatrist, perhaps better, will know that potentially that drug will work better in a specific patient, but based on the knowledge of depression in general. And so potentially ADHD, I will not reply directly to that question, but I would say in a patient that we think could have potentially some of those symptoms, the rationale, the approach will be this one, right? Because there's no specific drug that has been tested for that symptom in Huntington's. And I think just to finish, that's very important. You know, I think we're all very excited at thinking about the potential of disease-modifying therapies. But as we say, disease-modifying therapies, I mean, I think that the most realistic effect is that they will slow the progression of disease. So that means that symptoms will still exist, I think, you know, maybe later in the disease, maybe milder, which I think will be great. You know, instead of having maybe a comparison, you know, instead of having a common cold, or the reverse, instead of having a flu, you're going to have a common cold, much milder. But still, you need to have tools to address those symptoms in Huntington's disease. So I think it's still it's a area that we shouldn't forget ever.
Okay, let's go to a more general question now. So hypothetically, do you think is it easier to reverse Huntington disease progression or to prevent disease onset? Like, why aren't more studies being done on pre-symptomatic patients? Rachel, do you have any thoughts?
It's a tough question. I don't think there's really a concrete answer to that. I think, you know, our first aim with drugs is, you know, we want what clinicians and scientists would call a disease modifying treatment. So normally that means that you slow, halt, or reverse the symptoms of a disease. And any of those right now would be great for Huntington's disease. Probably the easiest thing to do would be to just slow things down, because what we don't understand yet is, you know, a lot of Huntington's disease symptoms come because, you know, brain cells are dying over time, and we have, you know, loss of brain matter. And so to reverse Huntington's disease, you know, you can imagine that probably you would need to replace those brain cells in some way or rebuild connectivity within the brain. And that's going to be really tough to do, I think, with current technologies. But that's where slowing things down or halting the rate at which, you know, you have brain cell death and other symptoms of neurodegeneration is probably the fastest way to go. And you're right, like it would be then, if that's your option, then it makes sense to treat people much earlier on in disease. I think the problem, and one of the reasons that hasn't happened so much, is that to put people into clinical trials, we need things that we can measure, and we need good biomarkers, and we need other kind of symptoms and metrics that we can score to see how people are doing when they take the drug or they take the placebo within a clinical trial. And for a while, that's been quite tricky with people earlier in the disease, but we have more and more research now about biomarkers, whether those are things that we measure in the blood or the spinal fluid, or whether it's from brain imaging, that we can really start to understand what's happening at earlier stages of disease. But my understanding as well, Tiago, you could definitely comment on this better than me, is that generally, like, regulatory agencies are not very keen on treating people earlier in disease, and so there is a bias towards selecting people later on. Is that right?
I think it's a little bit what you've just said, Richard, right? It's more a question of pragmatically how you show that the drug is working in someone that doesn't have symptoms. So it's really, you know, all the development of biomarkers that would tell either that the drug is doing what's expected to do, and then actually, you know, slowing down or preventing the, you know, the biology of Huntington's. So but actually, you know, I think I was very surprised. One of the trials that likely Blair Levitt talked about, the Pivot HD trial. So yeah, actually, so it's kind of more a question of let's say, and you know, for example, one outcome. So one thing we measure to see an effect could be, are people that are on the drug, are they getting a diagnosis later in their lives, for example, what we can call phenoconversion. And so in this trial, what was done, and I think is fascinating to see how it play out, is that so they used a prognostic measure that's called prognostic index HD, which tries to select subjects that we think by that algorithm that they are closer to have the onset of more symptoms. And so I think that's kind of a first step to potentially allow people that don't have symptoms to be part of these trials. But then it's, I think, ultimately, we like to intervene earlier, the earliest possible, right? And so it's, in a way, it's a contradiction, right? We don't have the tools, but definitely if we had the tools, that would be potentially the time where neuronal loss is less, so more number of neurons to rescue. So the potential for, let's say, stop the disease or really slow down the disease, it's much higher. So I think hopefully we'll get there.
And I think this is, comes back to what we were discussing earlier about these natural history studies and things like enroll and HD clarity and these other observational studies, which can really help us work out how we can track the disease at these much earlier stages, where you might not have any motor symptoms yet, but there are other things going on that we could probably measure and figure out and help track the disease over time. And one of those things is the NFL biomarker. So this is a small protein that you can find in spinal fluid. And we know that even at very early stages of disease, you can see an increase in the levels of the NFL protein. And this is, gives people hope that by measuring things like this and other biomarkers in earlier stages, that we might be able to start tracking how drugs work in people at earlier stages of the disease. But I know this is a question that comes up a lot, and it's definitely something that everyone wants to do. It's just figuring out how we're going to do it.
During the question, or the answer period, somebody asked, just for a brief definition of pre-manifest. What do we mean by pre-manifest disease? Mahmoud, would you like to take that one?
Yeah, I can take that, although I think Tiago might be in a better position to refer to the clinical definition. But essentially, when we generally talk about pre-manifest, is somebody who carries the mutation, but is not yet showing or has not been clinically diagnosed. I think, also it's even, for scientists and research, this is also can be a little vague, or, or we kind of have to specify what we mean by manifest in this case, because there is a range of symptoms, psychiatric, behavioral, and then there is motor. Some are easier to quantify, others could be having a bad day type situation. And so I think pre-manifest and a lot of what's been done in HD really refers to motor onset. But Tiago, please correct me.
No, that's absolutely true. I perhaps will just add something that, and many of you have heard, again, there's been an effort to try to maybe get out of this nomenclature, because again, it can have different meanings for different people. And some of you have heard of a staging system for Huntington's. And again, this is another example of how observational studies really helped and drove this research. So I think it ties a lot with different things we talked already. So observational studies. So how can we use markers to kind of define what we call a stage beyond any clinical symptoms or clinical features? And so this staging system basically goes by numbers. So 0, 1, 2, 3, and 4. And so really, and each stage really depends on specific testing, right? And ultimately, what could be a more, let's say, a clinical diagnosis, it's really based on the impact. So if there's any functional impairment on the individual that, of course, has a genetic mutation, then it's kind of this stage. But so it tries to quantify and be more objective of why someone would be, included in one stage versus the other. I have to note that this is still a research staging system, okay? But really permeates with clinic, right? Because as we start doing clinical trials, and already they're being done that take in consideration the staging system, I think anyone that's keen in taking part in these trials would like to know what's my stage? Do I qualify ultimately for a clinical trial? So I think it's something that it's a nomenclature that you're going to hear more and more in the future.
I have another sort of clinical question you might be able to answer, Tiago. Somebody was asking, can you speak to how spouse involvement could help? Like, for example, for the blood or DNA donation or participation in the studies, what can spouses do?
Sorry, what
How can a spouse help? Like when we have a spouse of an HD patient, when they're like maybe involved in Enroll HD, what can they do to help?
Yeah, I mean, I think in many ways, I see you always as a team, so as a duo that's taking part in research. And so to start with, there's all the care of that, a spouse can take part of, and maybe this immediately reports to perhaps when patients have more symptoms. But even at the beginning, when there's a disclose of a diagnosis, right? I think it's something that will be perhaps easier to do if you have someone that you can share that result with, and kind of leave that experience together. And when I say a spouse, I would say any significant other. I would extend it to that, so not be that restrictive. But then I think I would have also, and think others will have other perspectives and ideas. But then from a research perspective, the fact that we are able to collect data on people that we know don't have the mutation allow us to really understand what are signals that are specific of Huntington's versus what I would call noise, meaning that really is not specific of Huntington's and you couldn't find it in someone else, with the same gender and the same age. And so, by collecting data, clinical data, but also donating biological specimens, that's crucial, right? So that we can understand exactly what's specific in Huntington's in all these studies.
So how many companies total are working on HD therapies? Rachel, Tam, could either of you maybe expand a little bit on this?
It's an excellent question. So we know there's obviously loads of companies that currently have drugs that are in the clinic right now as we speak, which is great. There are companies that are, revving up to get started in the new year in 2023. We know that Roche has their generation HD2, which we're hoping is going to start in the next sort of 2 to 3 months for enrollment. So that'll be interesting to see how that goes. But we were actually going through this earlier before we got started on this discussion. And I think I counted over 30 companies who have stated that they are involved in Huntington's disease research in some capacity, which is really amazing. So this counts things all the way through from like people who are developing these like cool CRISPR gene editing technologies in, these startup biotechs, all the way through to these really big, massive pharmaceutical companies who have tons of money for research and development, like Novartis and Roche Genentech as well. So there's a really good breadth of companies involved doing all sorts of different angles of Huntington's disease research. Oh, Tam, we can't hear you. Hang on.
How about now?
Perfect.
Excellent. There's a pesky button on my headphones that gets unpushed sometimes. What I was saying is, I think we were all a little bit happy and surprised at how many companies are involved. And I would just add one other thing, which is that those are only the companies that have disclosed that they're working on Huntington's disease. So there may be even more. And like Rachel said, they're at all different stages of the clinical trial process. So some might be very early, but we welcome them.
Yeah, and it's important to note as well, some of these companies are trying to tackle Huntington's disease from many different angles. So we know that, like, for example, Roche recently announced at the Huntington study group meeting last week that not only are they going to be running their generation HD2 trial, which is going to try and find a better dose and a better dosing regime for the tominersen Huntington lowering drug, but they're also going to be trying to develop an Huntington's disease allele specific ASO treatment, which will not hit the normal protein, will only hit the harmful protein. And they want to develop their own splicing modulator, which is the same kind of drug that Novartis and PTC Therapeutics are also in the running for developing. So there are some companies, it's not like they've just got one thing on the go. They've got lots of different resources and different strategies that they're trying even within one company.
And I think like for those of you who saw Dr. Blair Levitt's talk, or maybe if you missed it, one of the really nice things that he said at the end was he explained how his wife is an oncologist. And he said, you know, she gets to cure her patients all the time because there's they've figured out over the years in cancer how to use multiple different drugs and give people like a regimen of drugs. And he said, you know, we're not there yet with HD, but we will get there. And when you look at the number of companies that are using all of these different strategies, you can see how, you know, if one thing helps a little bit and then another thing helps a little bit more, and some things for some people, like, you know, the wave allele specific drug might only work for 30% of the population, but the other 70, yeah, math is hard. The other 70% will benefit from the other approaches.
Yeah, absolutely.
So I've got a question now that I think each of you might be able to answer, or you might share the same answer. And I'll ask maybe Mahmoud to start first, but what do you think is the most exciting or promising approach for a new treatment?
That's a tough question. I think I'm still excited about the Huntington lowering efforts. There's been some setbacks, but I think, you know, as Dr. Levitt discussed, and it's been discussed in other forums, some of these might be just logistical or dosage or delivery issues. I think the mechanism, the idea that, you know, removing the mutant protein still is the most appealing to me. And it's just a question of whether we can do it selectively so that it's only the mutant copy that's targeted, and that the good, helpful copies sort of retained. And so, yeah, I'm still excited about this approach, although I do recognize that, again, there are really still big hurdles to overcome in achieving that goal. And then I think beyond that, you know, sort of the same idea, but now instead of antisense, using some of these splice modulators, you know, we might again be able to deal with the mutant protein, but using a small molecule that can be given to everyone without having to worry about what haplotype they have and what genotype they carry, and again with intrathecal injections, etc. All the other sorts of you know, sort of required challenges that need to be overcome with the Huntington lowering, you know, with the splice modulators, because it's these are all oral compounds. I think if we can do that, or if we can find one that's safe and that can accomplish this, then that would be my second most exciting approach, or maybe equally exciting approach.
As a clinician, I can resist. I think for me, those, as a clinician, that those would be quite exciting. You know, that you could just prescribe a pill or some liquid to a patient, you know, and that would be quite amazing, I would say. But I mean, having said that, and to my surprise, or maybe my bias initially, it's amazing first how patients, participants of trials are willing to, for example, do lumbar punctures. And to my surprise, that perhaps, you know, being in the Huntington's community, I was not that surprised, but I think it's so good that we can celebrate that all the time with each study. But actually, at least my personal experience, it was quite well tolerated, you know, and maybe, you know, in the last 2 years, I might have done, you know, maybe 70 lumbar punctures, and they were quite well tolerated. So, but ultimately, whatever, you know, really brings a you know, a positive effect, I think that will be a major breakthrough.
Is there, like, besides clinical trials, like, while waiting for one to take place, maybe, what else can people with HD do now to help advance research?
all sorts of things. I mean, I think like Leora said this yesterday, and she's much more eloquent than me, but I think her point was that just being here today and, you know, being interested in the research and learning, like this is its own form of advocacy, and you're learning as you're, well, I hope you're learning, and I'm hoping that we're giving you useful information that you know you have to arm yourself, but also that maybe you can share with other people, like within your family and your friends, and that's really, really important. But there's all sorts of ways as well that you can get involved in HD research, either as someone with the HD mutation, or if you do or don't know you haven't had, or have some of the trials, for example, don't actually need you to know your genetic test result. And so that's important to know as well. So there's all these observational studies that are currently recruiting. I think HDSA Trial Finder lists a lot of them and has lots of really great information. You can also find them on the FDA website and find out where clinical trials are happening and what's being done. But even just being involved in your local chapter at HSC, like that's a big thing, and finding your community and your network. So, you know, you don't have to be having a lumbar puncture every 2 to 3 months if you don't want to. You know, you just making connections with other people and staying engaged is pretty amazing as well.
Yeah, I agree completely. And I picked up on Leora's comment too, and it really rang true. It's just informing yourself and being able to communicate things about HD is its own form of advocacy. And, you know, like every little bit sounds cheesy, but every little bit helps. So any contribution, no matter how small, it makes a difference. And you can see it when you see over the years these major shifts that have happened, major changes. And so that could be, you know, by helping fundraise to bring the money in that's needed for the research. It could be like we were talking about how spouses can participate. I think Ed always says like be a wingman. So just being there to drive somebody to a clinical trial or, you know, support them in some way, that's contributing too. So there are lots and lots of things that can be done.
We have time for maybe a couple more questions. If anyone wants to raise their hand, you know, in the chat and ask a question personally, that'd be really appreciated. But if not, a couple more minutes, so I'll just jump to another one here. Somebody asks, because you talked about inflammation earlier, where do nutrition, exercise, and your general lifestyle come into play? And in what way can pre-symptomatic people optimize their epigenetics in this way? Anybody?
Tiago, we're looking at you again as a expert clinician.
Yeah.
Then we have to add a link with maybe more basic research. But I think my personal perspective is, in someone with Huntington's or someone, I would say, with neurodegenerative disorder, that disorder will perhaps have less impact in a healthy body, or someone that has, and that will, I say, with a proper nutrition, with exercise, as long as it's safe. So those elements, I think we recommend it. And if you learn in neurodegenerative disorders that it's important, right? And so promotion of mobility, a good cardiovascular health, and ultimately a good nutrition also. I think it's general recommendations that I would make to my patients. And so I'm not so sure about the direct link, for example, with Huntington pathophysiology, but again, it brings more that it's a person, so it's not just Huntington, and it's not just the brain, and so all these I would fit into a healthy lifestyle. Definitely something that someone with Huntington should have in consideration.
I would just add, I know that there have been a number of studies that look at, exercise and the Mediterranean diet. And I can't really speak to, I don't know how large they were or, whether how concrete the findings were. But one of the other things that comes up in terms of lifestyle is that stress can have a negative impact. So, you know, there are ways of dealing with stress. You know, there's behavioral cognitive therapy. You know, I don't know that there are any studies that prove that that's helpful in HD, but these are all things that certainly would be worth talking about, you know, with your physician or your social worker, or anything to reduce stress and increase your general health. We all know that, you know, eating well and keeping active is good for us, and we should all do that whether we have the HD gene or not.
Okay. So on that note, I think we're starting to run out of time. We might have 1 or 2 more minutes. See if there's any questions. Do you miss that? I don't think so. So I think now at this point, I'm just going to have to say thank you so much to this dedicated group of scientific volunteers. You guys do so much for the research arm of HSC and the HD research field. Right now, it's time for our final community awards and then the closing keynote on chronic resilience, which take place at 4:45 Eastern. So I'd just like to say thank you again one more time, and I hope to see you all there for our final keynote.